All right, welcome to the deep dive. Today, we're going deep on something, well, kind of fundamental, right? How we actually know what we know,
Mm-hmm.
especially when we're talking about, science.
Mm.
And that's where this idea of scientific reproducibility comes in.
Yeah, makes sense.
So for you, the learner, this is more than just, some academic debate. It really gets to the heart of how we build trust in, well, knowledge, especially when science is the source.
Absolutely.
It's a bit like peeking behind the curtain, seeing how the magic trick is done.
Yeah, I like that analogy.
So to help us on this deep dive, we've got some seriously thought-provoking stuff from the Stanford Encyclopedia of Philosophy. Think of it as our philosophical guidebook to this whole complex issue.
Sounds good.
Our mission, should we choose to accept it, is to unpack all the different things reproducibility can mean in science, why people are talking about a crisis, what's being done about it,
Yeah.
and most importantly, what all this says about the very nature of scientific knowledge.
Yeah.
Taking that philosophical angle. And hey, get ready for some serious aha moments along the way. No PhD required. So where do we even begin with all this?
Well, let's start with what seems like a simple question, right?
Okay.
What does it actually mean for a scientific result to be reproducible? You'd think it'd be straightforward, but
Yeah.
our sources point out these terms: reproducibility, replication, even repetition. They can have slightly different meanings depending on, well, what kind of science you're doing. Like, if you're working with computer models,
Mm-hmm.
reproducibility might simply mean that someone else can run your code, use your data, and get the exact same output.
Like checking the math, basically.
Exactly. And then in that world, replication might mean rerunning that whole simulation or experiment from scratch.
So that distinction is important right off the bat. So if it's all about numbers, reproducibility is like, show your work, let someone double check.
Right.
But in a lab, like psychology or biology, replication usually means someone else doing the whole experiment again, new data and all.
Exactly. Even DARPA, the agency behind a lot of cutting-edge research, they make this distinction too. Reproducibility for computational stuff, replicability for actually redoing experiments out in the real world.
So for you, the learner, the takeaway here is that reproducibility isn't a one-size-fits-all concept.
Yeah, it gets even trickier because even when we talk about replicating a study, we could be talking about two different things.
Okay, lay it on me.
First, you could say a study is replicable in principle, meaning the methods, the step-by-step instructions are so clear that anyone could theoretically follow them.
Okay.
But then there's also this hope that if someone does follow those instructions, they'll get the same or at least very similar results.
Yeah.
And what our source really emphasizes is that a study can be perfectly clear in its methods, totally replicable in principle, and still, when someone else tries it.
It doesn't work out the same.
Yeah.
Like, imagine a recipe. You can have a perfectly written recipe, replicable in principle, right? But if you use, I don't know, slightly different ingredients, or your oven's temperature is off, the cake might not be the same, not replicable in outcome.
Right.
So just having a detailed method isn't some guarantee of the same result.
Yeah, and that makes it tricky when we're talking about these replication studies. Are we just saying someone followed the original method, whatever they found?
Or were we expecting them to get the same kind of result?
Exactly. It's subtle, but important to keep in mind.
Absolutely. And then, to get even deeper into this, the Stanford Encyclopedia lays out these three, I don't know, kind of fascinating accounts of reproducibility from different perspectives. And this is where, the philosophical stuff really starts to shine.
Yeah, the first one's from Stefan Schmidt, who focuses mainly on the social sciences.
Okay, social sciences. What's his take on why replication is so important?
Well, he argues that these replication studies, they serve 5 key functions in science.
5. Okay, hit me.
First, they help control for sampling error. So making sure that the original result wasn't just some statistical fluke because of the specific people they use.
Right, not just a random blip.
Exactly. Second, they help control for artifacts, making sure the result wasn't caused by something unintended in the study design.
Okay.
Third, they act as a check against, well, potential fraud.
Yeah, got to cover all the bases.
Right. Fourth, they let us see how generalizable the findings are. Do they apply to different groups of people, different settings?
Okay.
And fifth, they give us a way to actually verify the underlying hypothesis itself.
So it's not just about spotting mistakes. It's about building confidence in the findings.
Exactly. It's multifaceted.
That's a really useful framework.
Yeah.
So how does he think we can achieve all these goals What are the tools we can use when we're replicating?
Well, he points to 4 classes of variables that we can either change or keep constant when we're doing a replication.
Okay, 4 classes.
1, the information given to the participants. 2, the broader context, like participant characteristics, the physical environment. 3, how they recruit participants.
Right.
And 4, the specific ways they measure the thing they're interested in, the dependent variable.
So by tweaking these things, holding others steady, we can ask different questions about the original findings.
Precisely. For example, if you mainly want to check for that sampling error, Schmidt says, focus on changing the participant recruitment. Keep everything else as close to the original as possible.
Makes sense.
If you're wondering if the finding was just an artifact of the specific context, change the setting, maybe some participant characteristics.
Okay.
And he argues that these first 3 functions, controlling for error, artifacts, and fraud, they're best addressed through direct replication, sticking close to the original.
So direct replications are about confirming those initial findings. Are they solid? Do they hold up in the original context?
Right, but those broader questions like generalizability and the hypothesis itself, those are more in the realm of conceptual replication.
So like testing the core idea in a new way.
Exactly. To see if it generalizes, you might change the recruitment methods. To really test the hypothesis, you might change the information given, the context, even how you measure the outcome.
But you'd keep the recruitment pretty similar.
Yeah. And he acknowledges it can be tricky to isolate these changes perfectly.
So to sum up Schmidt's view, direct replication is about, did they get it right?
While conceptual replication asks, is the idea itself right?
That's a great way to put it. And he sees this whole spectrum of replication, from super direct to very conceptual.
Yeah, each serving a purpose in building that scientific knowledge.
Absolutely. Now, the next perspective comes from Gomez, Jaristo, and Vegas. They took a very interdisciplinary approach, right?
Right, looked across a whole bunch of different fields.
So what was their angle on why these replications might differ?
They looked at 18 different disciplines, found a ton of ways, 79 to be precise, that a replication could deviate from the original.
Wow, 79.
Yeah, but they boiled it all down to 5 key areas where things tend to diverge.
5 key areas. Okay, I'm intrigued.
1, the physical site of the experiment. 2, the specific experimenters. 3, the apparatus or equipment they used.
Okay.
4, the operationalizations, which is how they measure the concepts. And 5, the characteristics of the population they're studying.
Okay, so some overlap there with Schmidt's classes of variables.
Definitely.
And then, like Schmidt, they connect these differences to why a scientist might choose to vary them.
Right. Like if you keep all 5 elements the same, you're mainly looking at what they call conclusion validity. Is the relationship between the variables real, not just random chance or a fluke sample?
Okay, a strict repeat is about ruling out flukes. What if you start changing things up?
Well, if you change the site, the experimenters, or the equipment, you're shifting to internal validity. Were the results really caused by what they thought, or was it something about that specific setting, those experimenters, that equipment?
Okay, interesting.
Then, if you change the operationalizations, how you're measuring things, you're looking at construct validity. Does the effect hold up, even if you measure it a different way?
And changing the population gets you to external validity.
Exactly. How well do the findings generalize to different groups?
So each change you make in the replication helps you answer a different kind of question about the original.
Precisely. So both Schmidt and this interdisciplinary view, they provide us with tools for thinking critically about replication.
It's like a philosophical toolkit.
Yeah, and by consciously choosing what to keep the same and what to change, we can really target specific questions about, reliability, validity, generalizability.
Yeah, that's really cool. Now, the third account comes from Hans Rader, and he has a slightly different lens.
He distinguishes 3 types of reproducibility.
Okay, Rader's 3 types. Let's hear it.
First, there's the reproducibility of what he calls the material realization of an experiment.
Material realization.
Like, he gives this example where 2 scientists might do the exact same physical actions to measure, say, the mass of an object.
Yeah, okay.
But one interprets the results through Newtonian physics, the other through Einsteinian physics.
So same actions, different theoretical frameworks.
Exactly. Same procedure, different meaning. But our source points out there's some ambiguity here.
Oh,
What exactly makes one material realization a reproduction of another?
right, because no two physical repetitions will ever be perfectly identical.
Exactly. And that raises some deep philosophical questions about sameness in experiments.
That's getting pretty meta, huh?
It is. Then the second type is reproducibility given a fixed theoretical description.
Okay.
So here, the interpretation is assumed to be the same, but the actual material realization can differ.
Okay, so flip it around.
Yeah. Like Ratter talks about studying social conformity. You could look at children in one study, adults in another, the idea peer influence leading to conformity that stays the same, but the participants are different.
So is that really different from testing generalizability?
That's a good question, and one that Ratter's work makes us think about.
Like, are they separate things or just different sides of the same coin?
Right. And finally, there's what he calls replicability.
Okay, replicability.
This is where different procedures, even based on different theories, lead to the same result.
So different paths, same destination.
Yeah. He uses the example of measuring the boiling point of a fluid.
Okay,
You could use different types of thermometers, different mechanisms, but you get the same boiling point.
interesting. So different procedures can converge on the same truth.
Right. And Schmidt actually sees Ratter's second and third types as quite similar to his own concept of conceptual replication.
So testing the same idea through different methods or contexts.
Exactly. So across these three philosophical accounts, we see this recurring theme, distinguishing between strict repeats and more varied attempts to validate a finding.
Yeah, that's a key takeaway. Now, let's move on to something a bit more, well, contemporary. This reproducibility crisis, or as some prefer, the credibility revolution.
Right.
What's all the fuss about?
Well, these terms really gained traction in the last decade or so, mainly because of these large-scale projects that tried to replicate published findings.
Okay.
And the results were often, well, disappointing.
Oh, in what way?
A surprisingly low percentage of those original studies could be reproduced. It happened in medicine, life sciences, behavioral sciences.
Like, how low are we talking?
There was this big Nature poll in 2016. Over half of the scientists surveyed thought there was a replication crisis.
Oh, that's a lot.
It is. But it's worth noting that some, like Samin Vazir, prefer credibility revolution.
Because it focuses on the positive changes and the increased awareness.
Exactly. But still, this whole thing, it challenges this assumption that published findings are inherently reliable.
So it's not that science itself is broken. It's more like,
It's a period of self-reflection, right?
yeah, like science is taking a hard look in the mirror.
Exactly. And this crisis or revolution, it's really a bunch of interconnected problems.
Okay, break it down for us.
First, replication studies are just rare in published research. Second, when they have been done, they often fail.
Like those large-scale projects we were talking about.
Yeah, exactly. Third, there's evidence of publication bias.
We'll get to that, I'm sure.
Right. Fourth, there are these questionable research practices that can inflate false positives.
Okay.
And fifth, there's often a lack of transparency in how research is reported, making replication even harder.
All of those sound like pretty serious challenges to, this ideal of self-correction in science.
Yeah, they really are.
Okay, let's dive into each of these a bit more. Starting with the lack of published replication studies. Why is that such a problem, philosophically speaking?
Well, in theory, replication is how science is supposed to self-correct.
Yeah, you build on previous findings through independent verification.
Right. But if we can't verify those claims, how can we be sure we're building on solid ground?
So how little replication is actually happening?
In some fields, like psychology, it's as low as 1%. In ecology and evolution, it's even less.
That's a far cry from the ideal.
It is. And it makes you wonder, what's discouraging this crucial part of science?
Right. What are the incentives at play?
Well, historically, many journals prioritized new, exciting findings over replications.
So replications were seen as less impactful, less interesting.
Yeah, and funding agencies tend to favor groundbreaking discoveries, not the meticulous work of checking what we already think we know.
It's that pressure to be novel, to advance your career, and it might actually be hurting the reliability of scientific knowledge.
Precisely.
It's a tough balance. And then we have the issue of these large-scale replication projects where a lot of studies couldn't be reproduced.
Right.
Let's start with the reproducibility project. Psychology. That one sparked a lot of debate. What did they find?
It was a huge undertaking. Hundreds of researchers tried to replicate 100 studies from top psychology journals.
Ambitious.
Yeah. And only a minority were successfully replicated.
Like, give me the number.
Only about a third of the replications got the same level of statistical significance.
That's concerning. But I remember there were some criticisms of the project itself.
Yeah, some argued that the replications weren't close enough to the original studies.
Yeah, okay.
Maybe the samples were different, or there were subtle variations in how they ran the experiment.
So the failures to replicate might not mean the original findings were wrong.
Exactly. But the researchers behind the project pushed back. They said they often consulted the original authors,
Okay.
and they found that the perceived similarity between the original and the replication didn't really predict whether it would be successful.
Interesting.
There were also some Bayesian reanalyses of the data. They suggested that while the evidence for replication might be weak in a lot of cases, it wasn't proof that there was no effect.
Ah, so the interpretation of the results gets complicated.
Right. Different statistical frameworks can lead to different conclusions.
So even with the criticisms, this project was a wake-up call for psychology.
Definitely. It made people think hard about their methods and the reliability of their findings.
And what about other fields? I remember you mentioned some challenges in cancer biology.
Yeah, the Reproducibility project. Cancer biology faced even bigger hurdles.
In what way?
Well, a lot of the original papers just didn't provide enough detail.
So it was hard to even design a proper replication.
Exactly. It was a huge problem right from the start. They ended up doing far fewer replications than planned, and what they did find suggested low reproducibility in preclinical cancer research.
That's a big deal, especially for something like cancer research.
It is, and it raises questions about the standards of reporting in science.
Well,
If we don't even know how a study was done...
how can we verify it?
Exactly, and that lack of transparency seems to be a problem across different fields.
Undermining the whole idea of open inquiry.
Yeah, it really does. Now, the source also mentions the Many Labs project, which focused on replicating classic psychology findings across many different labs.
Okay, so a different approach.
Right. They got more consistent results for some well-established effects, but they also saw a lot of variability in the effect sizes.
So even seemingly solid findings can be influenced by the context.
Precisely.
And then there are those reports from biomedical research.
Right. Quite low reproducibility rates in those, with huge implications for medical advancements.
And huge costs, too.
Billions of dollars annually.
Wow.
Yeah, it's a big problem. But it's not all doom and gloom. The experimental philosophy project, for example.
Right, they had a much higher reproducibility rate.
Yeah, something like 70%, which is way higher than some other areas.
Why might that be?
Well, maybe it's because their questions and methods are simpler, more focused on basic intuitions.
Okay, so the experiments themselves might be more robust.
Yeah, less susceptible to little variations. And it suggests that the reproducibility challenges might vary a lot depending on the field.
Yeah, it's not one size fits all.
Right. And the Social Science Replication Project, which looked at studies from Nature and Science, they also had higher success rates, which suggests that maybe those journals have stricter standards.
So the takeaway here is that reproducibility isn't a monolithic issue.
It's complex. It depends on the field, the methods, the questions being asked.
Okay, so we've talked about the lack of replication studies and the mixed results for the ones that have been done. What about publication bias?
Right, that's the third point.
Why is it such a big deal, philosophically, that there's this bias towards publishing only positive results? You know, those that show an effect.
Well, ideally, the scientific record should be objective.
Yeah.
But if only the studies that show an effect get published, our understanding of the world gets distorted.
Like only hearing about the wins, never the losses.
Exactly. And this bias runs deep in scientific publishing.
Okay, how so?
There's this assumption that statistically significant findings are more interesting, more publishable.
And no results are seen as failures.
Yeah, even though they can be just as informative.
Like knowing something doesn't work can be just as valuable as knowing it does.
Exactly. It's a misunderstanding of how scientific evidence works.
So how bad is this bias, really?
Well, there's this work by Daniel Finelli that looked at the proportion of statistically significant results across different fields.
Okay.
And it was incredibly high, often way more than you'd expect, given the typical statistical power of studies in those fields.
Statistical power, just to remind everyone.
It's the probability of finding a real effect if there is one.
Right. So if studies generally have low power...
You wouldn't expect so many significant results unless there was a bias.
Ah, so this excess significance is a red flag.
Exactly. It suggests that null results are being swept under the rug.
And that brings to mind Rosenthal's file drawer problem. All those unpublished studies hidden away.
Right.
How does that play into this whole issue?
Well, meta-analyses, which try to combine findings from multiple studies,
Yeah.
they usually rely on the published literature.
Yeah.
But if a bunch of null results are never published...
The meta-analysis is based on incomplete data.
Exactly. It can make effects seem stronger and more prevalent than they actually are.
So it's not just a problem for individual studies, it can affect our whole understanding of a topic.
Precisely. It highlights the danger of relying solely on what's published and the importance of trying to account for the missing evidence.
It's like trying to understand a story with half the pages ripped out.
Yeah, that's a good way to put it.
Okay, so this bias towards positive results creates this pressure to find something significant.
Right.
And that pressure can lead to, well, questionable research practices, QRPs.
Exactly.
What are QRPs and what philosophical issues do they raise?
They're behaviors that, while not outright fraud, can still make it more likely to get those statistically significant results,
Okay,
even when there's no real effect.
like bending the rules a bit.
Yeah, something like that. And philosophically, this raises serious concerns about the trustworthiness of research.
Yeah, if you're manipulating the process to get the results you want.
It undermines the whole point of science.
So what are some examples of these QRPs?
Well, our source gives us a helpful table.
Okay, let's see.
One is selectively reporting only the outcomes that were significant.
Cherry picking the good stuff.
Right. Another is collecting more data until you get a significant result.
So keep going until you hit the jackpot.
Yeah. Or rounding p-values down just below the threshold.
Fudging the numbers a bit.
And then there's excluding data points after checking if it affects the significance.
Okay.
And arcing, which is hypothesizing after the results are known.
Ah, so you make it look like you predicted it all along.
Exactly.
And these practices, they might be happening consciously or...
Or unconsciously. It can be subtle.
But the point is, they can lead to false positives.
Right. There was this study by Simmons, Nelson, and Simonson. They showed that even small amounts of flexibility in how you collect, analyze, and report data can hugely increase the chance of getting a statistically significant result just by luck.
So it's not necessarily intentional deception. It could be unconscious bias.
Yeah, but the pressure to publish to get funding to advance your career.
It creates these perverse incentives.
It does. It highlights this tension between wanting to do good science and the pressures of the system.
And then the fifth point is the lack of transparency in reporting.
Right.
Why is that such a problem philosophically?
Well, transparency is key for science to be open and self-correcting.
Right.
If researchers don't give enough detail about their methods, their data, their analysis, it's almost impossible for others to evaluate their work, try to replicate it, or build on it.
So it blocks the self-correction process.
Exactly. And there's research showing that reporting practices are often inadequate.
Missing details, unclear procedures.
Yeah, and this can happen for different reasons, maybe space limitations in journals, assumptions about what everyone already knows.
Yeah.
But it can also be a consequence of QRPs.
Like hiding the messy details.
Yeah. So it's a catch-22. The pressure to get significant results can lead to less transparency, which then makes it harder to verify those results.
A vicious cycle.
It is.
Okay, now let's shift gears and talk about meta-science.
Okay.
This idea of science studying itself.
Right. It's a fairly new field, but it's growing fast.
So what exactly is meta-science and what's its role in all this?
It's basically using the tools of science to examine science itself.
Okay.
Like, how is research conducted? How is it reported? How are findings evaluated? What are the incentives?
So it's about understanding the process of science, the good, the bad, and the ugly.
Exactly. And it's driven by this recognition that we need more rigor and self-awareness in science.
And it's not just about identifying problems, it's about finding solutions.
Right. A lot of those reproducibility projects we talked about, they're examples of meta-science in action.
They provide data on how big the problem is and where we need to focus our efforts.
Exactly. And meta-science also looks at things like publication bias, questionable research practices, how well researchers understand statistics.
So it's a data-driven approach to improving science itself.
Precisely. Holding science to its own standards.
Love it. Now let's get into some of the deeper philosophical stuff. The source mentions the experimenter's regress.
Ah, yes.
That sounds pretty heady. What is it, and what does it say about scientific knowledge?
It's this philosophical problem that arises when we're trying to figure out if experimental results are reliable.
Okay.
It starts with a simple question. How do we know if our equipment is working right? And how do we know if the results are accurate?
And the problem is, those two things are intertwined.
Exactly. To know if our results are accurate, we might compare them to results from equipment we think is reliable.
But how do we know that equipment is reliable?
Right, usually because it produces results we expect or that align with other findings or theories.
But that creates a circularity.
It does. The accuracy of the results depends on the reliability of the equipment. But the reliability of the equipment seems to be based on the accuracy of the results.
So it's a regress.
Exactly. It challenges this idea that we can have perfectly objective verification.
And how does this play into replication?
Well, imagine one lab tries to replicate another lab's findings, and the results conflict. Was the original finding wrong, or was the replication not done properly?
And figuring that out is where the regress comes in.
Right. What makes a replication proper? Did they really recreate all the important conditions?
Especially given that scientific papers often leave out details, and a lot of expertise is tacit knowledge.
Precisely. Collins, who developed this idea, points out that papers can't include every single detail, and a lot of skill is in the know-how, not the written instructions.
Right.
So it can be really hard to know if a replication truly matched the original.
And that can lead to arguments about who's right.
Exactly. And Collins suggests that sometimes things other than objective evidence, like social networks, prior beliefs, career interests, can influence which interpretation wins out.
So social factors can sometimes play a role in science.
That's his argument, and it's certainly controversial. Some, like Alan Franklin, strongly disagree. They emphasize the strategies scientists use to validate their results.
Like checking their instruments, eliminating errors, looking for independent evidence.
Right. And he argues that these strategies show that there's more to it than just social negotiation.
So it's a debate about how objective science really is.
It is. But even if social factors do play a role, it doesn't mean all science is subjective.
Yeah, it's more nuanced than that.
Right. And there's this work by Odysseus Feast who looked at the Mozart effect.
Okay.
She argues that disagreements about replication often come from evolving concepts and assumptions.
So the process of replication can actually help refine our understanding.
Exactly. The conflicting results in the Mozart effect eventually led to a more precise understanding of when the effect supposedly happens.
So the regress highlights a challenge, but it doesn't mean replication is useless.
Right. And there are ways to navigate these challenges through discussion, better methods, and looking for converging evidence.
Okay, so now the source asks, is replication the defining feature of science?
It's a good question.
It's often said that reproducibility is at the heart of the scientific method.
Right. And there's some strong arguments for that.
Like,
Well, by being transparent about their methods, scientists allow others to test and potentially verify their claims.
that's the self-correction mechanism.
Exactly. It's how errors get caught and trust is built. Some even see reproducibility as a way to distinguish real science from pseudoscience.
So it's a core value of science.
It is. But there's another perspective, too.
Oh,
Friedrich Steinel argues that the importance of replication has actually varied throughout history.
okay, interesting.
He says things like how well the results fit existing theories, the difficulty of replication, and the credibility of the researchers all play a role.
So it's not a simple replication equals good science equation.
Right. He uses examples like high-temperature superconductivity.
Okay.
It was quickly accepted because lots of labs replicated it, even though it clashed with some theories.
Interesting.
And then there's von Guericke's vacuum pump experiments. They were accepted without much replication because they were hard to replicate, and he did these public demonstrations.
So context matters.
It does. Steinle's point is that we need a more nuanced understanding of replication.
Not a one-size-fits-all rule.
Right. And then there's Earp and Trey,